Hi, good morning, everyone, and welcome to this practical revision class. And for those that will be seeing my face for the very first time, my name is Calista Daniels, and I am your practical demonstrator for this section. And we are two, you'll be seeing the second person shortly after me. So I'm starting with, it's going to be a bit of instrumentation and theory because we know that for MBBS, your practical exam in physiology is mostly steeple chain pattern, identification of the necessary equipment and apparatus that are used to perform the individual practicals in physiology. So I'll be starting with the microscope. Yeah, I'll be showing you guys the equipment. I'm going to be as fast as possible so that we don't spend so much time. So you'll be asked, for example, you are asked to identify, like for the microscope, you'll be asked to identify it. So you should be able to know the name and the various parts and what they are used for. So for the microscope, I'll give you a few examples and what they are used for. So you'll find out, you Google the rest yourself. So let's say, for example, identify specimen A, and this is specimen A. So you can be asked to identify this. We'll just label it specimen A, identify this. So you identify and list two or three parts and their uses. So let's take, for example, identify specimen A, which is the microscope, and list two uses. Microscope is used for microscopy. It is used for histological examination and cutting. That is for the advanced microscope. This is a binocular microscope, by the way, the one I just showed you. And the parts of the microscope, we have the objective lens, we have the stage. This is the stage. This is the fine. We have the fine and the coarse adjustment. We have the stage. We have the light source. Okay, it's not connected. So we have the light source. We have the objective lenses. And that is it for it. So I just want to just do that very fast. So we'll move on to ROBC and WBC practical. So for the ROBC, you are going to be asked to identify the equipment and state their function or uses. And also you can be asked to list or state the normal values for male and female ROBC. ROBC, I mean red blood cell count or red blood cell. So you have to note this: the equipment and their uses, the normal value, international normal value for ROBC for male and female, and sometimes you could be asked for newborn. Yes. So let's start with. So I'm going to start by giving you the normal values for the male and female red blood cell. So for 4.5 to 6.0 million cubic millimeter. While for the female, it is between 4.0 to 5.5 million cubic millimeter. For the newborn, it is always higher than the adult. For the newborn, it is always higher than the adult. So I need to talk a little about the principles. So blood is diluted with ROBC diluting fluid, which is the Hayem's fluid. That is it. You can see. I don't know if you can see. This is the Hayem's fluid, and counted using the hemocytometer or the Newberg counting chamber. This is the Newberg counting chamber. This is the Newbar counting chamber. So I said, let me explain it again. Blood is diluted with the RBC diluting fluid, which is called the Hayem's fluid, and is counted using the hemocytometer or Newbar counting chamber under the microscope. I'm going to explain the principle. So for the equipment that are needed for RBC, we have the RBC pipette, which can easily be identified with this red bulb inside. I don't know if you can see it. There is a red bulb. Can you see? There is a red bulb inside the pipette for the red blood cell, and is graded up to 101. Yes. So these are the things you may be able to use to identify a red blood cell and pipette when you see it. And the tube for the sucking has a red mouth. So this is it. We have the Hayem's solution of fluid. We have the Newbar counting chamber, and we have the microscope. I've already showed you the microscope. So for steeplechase exam, you'll be asked to, let's take for example, identify specimen A. Let's assume this is specimen A. This is red blood cell pipette, and it's used for diluting the red blood cell with the diluting fluid in preparation for reading. So I hope you guys are getting the point. So I said I've listed the equipment or apparatus that we use for red blood cell. We have the Newbar counting chamber, we have the red blood cell pipette, we have the Hayem's solution or fluid. We use the microscope for it. So what happens is that you draw blood to the mark of 0.5. You draw blood to whole blood to this mark 0.5. I don't know if you can see it, but if you can't, it's okay. 0.5, and you suck up the diluting fluid up to the mark of 101, and you gently mix. You gently mix by gently rolling or rocking between your palms. And before then, you must have charged your Newbar counting chamber. What do I mean by charging it? You get the cover slip. You can put a little bit of use the magnetic spirit to clean it, make sure it's clean, and you place it on top of this place, the groove. Yeah, you place it on top of the groove, and you gently apply, gently put in the already diluted blood, and you place under the microscope, and you read with by 40. Yes, you read with by 40. So I see. Okay. So that is it for the red blood cell. And also you have to have at the back of your mind that red blood cell and white blood cell have similar principles and similar apparatus, but the only difference between them is the pipettes. So let me show you the difference. This is the white blood cell pipette. You can see the white blood cell pipette has a white bead right inside, and the tip is white also, and the grading of the scale is different. So for white blood cell, you have 0.5 and 10. 0.5 and 10. While for the red blood cell, you have 0.5 and 101. So you can see, let me put it hold so you can see the difference. So you see the difference. See the difference? Let me show you. Can you see? So that is the difference. And it's the same pattern of filling up the Newbar counting chamber and same way to read. So I want to talk about, and the fluid for white blood cell is duct solution. tox solution. Tox solution. So that is the difference. You see the difference. See the difference. See, this is IM solution for red blood cell. This is tox solution for white blood cell count. So I want to give you the value for white blood cell. The white blood cell is normal for both, it's the same for both male and female. I will have between 4,000 to 11,000 cubic millimeter. I said again, 4,000 to 11,000 cubic millimeter. And there are certain conditions that can cause increase in white blood cell, and there are situations that can cause an increase or decrease in red blood cell. So I'm going to start with red blood cell. So for red blood cell, dehydration, high altitude, and or lung disease, polycythemia, can cause an increase in red blood cell. I don't know if you get that. Dehydration, high altitude, or polycythemia can cause an increase, and there are numerous other factors or other things that can lead to an increase in red blood cell, but I just want to give you this few so that we can keep for time's sake. Why for decrease? A decrease can result from anemia when you are anemic, or there's blood loss, or there's nutrient deficiency. It can lead to a decrease in red blood cell. Do you get that? Okay, so let's continue. An increase in white blood cell indicates infection, inflammation, or stress, leukocytosis, whereas decrease in white blood cell can increase in certain conditions such as infection like bacterial, viral, fungal infections. This can lead to an increase in white blood cell. We have inflammation such as tissue damage, autoimmune disease, lupus, allergies. These are some of the examples of inflammation that can cause an increase in white blood cell. We have stress, physical injury, extreme emotional stress, or strenuous exercises. Example of stress that can cause increase in white blood cell. We have medications, smoking, steroid, and smoking can cause an increase or a raise in the count. We have leukemia. Leukemia is another reason why your blood cell count, white blood cell count, will be high. Yes, will be high. So let's talk about leukopenia, which is decreased decreased white blood cell. We have the bone marrow damage. When there is a damage to the bone marrow as a result of chemotherapy, cancer, or radiation treatment. We have infections such as severe or viral infection, HIV, hepatitis. This can cause a decrease in your white blood cell. Autoimmune disorder, nutrient deficiency like low folate or vitamin B12. This also can cause a decrease in white blood cell. We have medication such as drugs, some certain medication like chlorophenicol or phenytoin. Yes, these are some of the medications that can cause a decrease in red blood cell. If you have any question, please you can drop that in the comment section. I will be glad to answer them. So you can drop your this thing. And if you haven't subscribed at this point, please make sure to subscribe to my channel. So let's continue. So we are through with RBC and red blood cells. So I'm going to talk about packed cell volume. So the next thing I'm going to talk about is packed cell volume. I'm going to discuss packed cell volume, PCV, ABO blood grouping, and hemoglobin estimation together. I'm going to take them.
Good day, guys. Good day from wherever you are watching. My name is Olamide, and technologies from Physiology lab,
BSc, MSc Physiology.
Unimed, Unimed Undo. So today we'll be talking about ESR, which is a erythrocyte sedimentation rate, and then PCV, packed cell volume. We also call it hematocrit. Yes, hematocrit. That's cells in the blood, the volumes of cells in the blood, the living component of the blood. We have the ABO, blood grouping system. We're discussing about that, and the hemoglobin estimation. Now, the objective of this revision is to determine the erythrocyte sedimentation rate. How to determine the erythrocyte sedimentation rate? Because this is a revision in form of alternative to practical. We are not going to carry out the procedure. We just want you guys to be familiar with, because we've already done the procedure in the laboratory. So we want you guys to be familiar with how it's been done once again. So, and then we're going to talk about the principles of RBC grouping and then hemoglobin estimation. Now, talking about ESR. ESR means the rate at which the erythrocytes in the blood, that's the red blood cells, the way this rate at which they sediment, that is the way they settle. When you have a blood sample and it is placed vertically. Now we have an equipment we use. We call it the Western Green ESR rack. Western Green ESR rack and two Western Green. So this is how it looks like. Inside the ESR Western Green ESR cup, there is an anticoagulant, sodium citrate. That's the anticoagulant inside the Western Green ESR cup. Now, there's a proportion of this anticoagulant that is already calculated to the volume of blood that will be ejected into this cup. Now, during this procedure, we have to take blood sample from subjects using the needle and syringe and introduce the blood into this cup. Now, this is where the blood is ought to get to, the neck part of this cup. That's where the blood sample ought to get to. Now, there's already anticoagulants that are appropriate for that volume inside this cup. So the importance of the anticoagulant is to prevent the blood from coagulating, because we don't need the blood to coagulate, we just need it to settle. So once that is being done, this cup, you can open this cup. You open this cup when you want to introduce the blood. You just open it like this and introduce the blood and cover it. Now the cup has a seal that is breakable. When we apply pressure, the seal will break and the tube will be able to enter into the solution. Now, to the presence of capillary pressure, to the help of capillary pressure, the blood sample will move to the tip of this tube. And now the tube is graded from 0 to 190 millimeter. So once this whole setup is standing like this, within 1 hour we check the rate at which the blood sample are sedimented, and that is the ESR of the individual. So it is usually calculated in rate of sedimentation per hour, so millimeter per hour. So that is the calculation, and the ESR is usually larger. The ESR is usually larger in female compared to male. It's usually larger in female compared to male. So reason for this is being that the male has blood of a male subject is more viscous, and the reason for the higher viscosity is the presence of large volume of cell, larger amount of red blood cell, white blood cell, and other cells, platelets and other cells present in the blood. So the three cells are the white blood cells, red blood cells, and the platelets or thrombocytes. So they are usually larger in male compared to female, which makes the male blood more viscous. And then the more viscous the blood sample is, the lesser the sedimentation rate. So the higher sedimentation rate, that is the reason for the higher sedimentation rate in the female compared to male. Now, this procedure, anything out of the normal in physiology, it is pathological, it is not physiological. So this procedure can help us to detect if an individual has low cells, lower number of cells compared to normal, which will lead to higher ESR compared to the normal ESR that is expected of that individual. So with this procedure, we can suggest if an individual has some pathological conditions like tuberculosis or any conditions that affect the cells of the blood, for example, HIV virus. So most of these procedures are just pre-test before the further test to actually determine what is actually wrong in that individual. Now, for the ESR in male is usually between 8 millimeter to 10 millimeter per hour, and that of female is usually from 10 to 12 millimeter per hour rate of sedimentation. Now, when taking this procedure, there are some precautions that we ought to take. Now, this Western green tube is a modern method, the present method in which we do ESR. The former method usually involved the Winthrop method, where we prepare the anticoagulant in the lab ourselves separately, and we do calculation. We calculate the proportion of anticoagulant we are going to mix with the blood, and the Western green has its own rack that enables us to stand the Western green tube inside the rack. But with this one, you can always stand this one inside a foam prep that is meant for this procedure. Now, the precautions is that you make sure that the setup is left vertical for an hour before recording it. Also, make sure that the anticoagulant is present in the cup, because of production error, you can end up finding a cup that does not have anticoagulant in it, and that will make the blood to coagulate, and once the blood coagulates, you will not get proper results. So let's move on to ABO grouping. Now, ABO grouping has to do with the method of grouping blood using the antigen antibody reaction. Now, this antigen antibody reaction is what is known as agglutination. Now, so in ABO blood grouping, it is based on the Land-Steiner's law, the presence of antigen A or B on red blood cells to determine the blood group. Now, we'll be talking about the ABO blood grouping system, which works with the principle of the Land-Steiner's law, which has to do with the reaction between antigens and antibodies. Now, for the ABO blood grouping, we have 3 antiserals, call them antiserals, that we use as reagents, reactors, as antigens for determining the blood grouping system, determine the blood group of individuals. This is anti-D, which is usually the resource reactor. Anti-D, the anti-D is for resource grouping. We have the anti-B, sorry, this is anti-B, the yellow color is the anti-B. Then we have the anti-A antiserals. Now, for this blood grouping procedure, we use blood well, but for practical sessions we use the white tie. This is a white tie. The white ties. So on the white ties, we are going to mark.
If you finish this one, you can come back. You can continue with.
Okay. We mark the white ties with a marker. We have A, B, and O. Now, A, B to represent the blood group A and B, and the O to represent the resource antigen, the results factor. So with that, we are going to have 3 drops of blood from each subject, one subject at a time. 3 drops of blood, a drop of blood on A, a drop of blood on B, and a drop of blood on D. So we will now introduce anti-A on the drop of blood labeled A, anti-B on the drop of blood labeled B, and that for D too. So what we are looking for is the agglutination reaction. Any drops that shows agglutination, that means the blood groups there matches. For example, if there's agglutination on A, then the person is A positive. It's A, sorry, not positive, yes, A. If the person has or the subject blood has agglutination on B, the subject blood is B. Now, to know whether the subject blood is positive or negative, that has to do with the Rexus factor. That means either Rexus positive or Rexus negative. Now, if the subject blood has agglutination on A and then has agglutination on O, then the blood is A positive. But if there is no agglutination on O, which is the results, then the blood is O negative. So whether a blood group is positive or negative depends on whether there's agglutination on the rhesus antigen. So for the blood group to be positive or negative, that means results positive, results negative. Now, if a blood sample has no agglutination on 3 of them, A, B, and O, no agglutination, that blood sample is O negative, and that blood sample is a universal donor blood. That has no antigen whatsoever. So that individual can donate to all the blood groups we have available. So that individual is known as a universal donor. If a blood sample does not have agglutination on A and B, but there's agglutination on D, that blood sample can be donated to every other blood sample that is rhesus positive. You can donate it to AB positive, O positive, A positive, B positive. That blood sample is not yet a universal donor because it cannot donate to any blood group that is rhesus negative. So packed cell volume, that's the total volume of blood occupied by red blood cells. And the principle involves heparinized blood is usually centrifuge. Now the reason why we use heparinized blood, that is an anticoagulant. In this procedure, the blood sample is meant to be centrifuge and not coagulate. So blood sample is usually taken from the subject and introduced into an heparinized capillary tube. This is sample of this is a plain capillary tube. This is an heparinized capillary tube. Heparin is an anticoagulant. So usually introduced into the heparinized capillary tube. You can see the red lining at the top signifies that this is an heparinized capillary tube, whereas the plain capillary tube is usually blue at the tip. Now, you see, when the blood is introduced into this capillary tube, the capillary tube is usually placed horizontally like this at the tip of the finger where the blood is usually taken from after pricking the subject with a sterile lancet blade. Now, we make sure that the capillary tube is filled to 3/4, 3/4 of its total quantity, and then one portion of the tube will be closed with plasticine. This is plasticine used to occlude close an end of the capillary tube. After that, the capillary tube is then introduced into a centrifuge. We call it microhematocrit centrifuge. This microhematocrit centrifuge has wells that can take the size of a parenized capillary tube, the size of a capillary tube. So once the capillary tube containing blood, which and one end of the capillary tube is already sealed with plasticine, is placed inside the centrifuge. The capillary tubes are placed in the well at opposite direction to balance the well, and then you lock it up, and then you centrifuge at 3000 revolution per minute for 5 minutes. Once you are done, you will see the different layers. You see the first layer you are going to see is the hematocrit, packed cell volume. You can call it packed cell volume or the hematocrit. Then the next layer you are going to find is a tiny buffy layer. Then the serum or plasma is left at the top. Then with that, you bring it into the microhematocrit reader. This is the microhematocrit reader for reading the PCV. So there's a way to place the capillary tube inside the microhematocrit reader. The portion where the capillary tube is sealed with plasticine. The portion where the capillary tube is sealed with plasticine aligns with the zero marking, and the portion where the blood or the fluid, the total fluid inside the capillary tube gets to is going to align with the 100% marking on the microhematocrit centrifuge. Then the next thing is to look for is to align the ruler at the tip, at the meniscus of the PCV, the hematocrit, and then get the recording, the actual score for the PCV. And then there's normal PCV for male and female subjects. Normal PCV for male is between 40 to 54%, because PCV is calculated in percent, and then that of female is between 37 to 47%, and 50 to 60% in newborn. Why do we have higher PCV in newborn? Because newborns have higher red blood cells, and that's why newborn go through jaundice to break down these red blood cells to normal as they grow older. Now about the ECG. So the ECG is also known as it's a procedure, it's a physiological procedure. So it's a practical that relates with the cardiovascular physiology, which is the study of the heart and its pumps, heart and the blood vessels. That's the science of cardiovascular physiology. So ECG itself is known as, it's a short form of electrocardiography. Electrocardiography, which is the science of studying and interpreting the electric activities of the heart. Science of studying and interpreting the electrical activities of the heart. Now, there's an electrocardiogram, and there's electrocardiograph. Now, electrocardiograph is the machine. I would like to bring the machine to the screen. Electrocardiograph machine is the machine that is being used to record the electric activity of the heart. This is an electrocardiograph. This is an electrocardiograph machine that is being used to record or detect the electric activity of the heart. So it has a monitor. It has a monitor from what I showed you. It has a monitor. It has a screen where you can see the electric activity of the heart. Now, the machine does not work on its own. It's not working with internet or electrical motors without having wires. It has wires. It's not working with signals flying without connection. It has electrode. This we call it the ECG electrode. These are the electrodes. These are the electric wires that are connected to certain regions in the body, surfaces in the body, and they pick the electrical activities and they transfer it to the ECG as signals that can be seen as graph on the ECG machine or the electrocardiograph. Now, this result is also printed on a graph paper that we call the electrocardiogram. Once it's printed on the graph paper, it is seen as electrocardiogram. That is the recording that is being shown on the graph paper, electrocardiogram. So, how is this procedure done? The practical aspect, how do we do it? Now, I've talked about a few equipment that we use for this procedure. I've shown you the ECG electrode, which I'm still going to break down and discuss more about. I've shown you the ECG machine or the electrocardiograph. Now, there's gel, the ECG gel, which is a conductive gel that's applied to the surface in which the electrode is going to be attached to. And the major thing you need to know as a physiologist and medical practitioner, before we talk about the interpretation, which is something you advance to learn because it's not a part of the curriculum at this level. So at this level, you need to know about how to place the electrode and how to do the recording, the procedure of the ECG, not the interpretation. Now, I'm going to show you or tell you, discuss with you, explain to you how to fix the electrodes, which is very important. So there are two or four parts where the electrode are placed. We have the chest region. I'm going to classify them as region. We have the chest region, that's just the two regions, and the limbs, upper and lower limbs. Now, placing of electrode on the chest, we have 6 chest electrodes that are usually in form of bulbs like this. 6 chest electrode, V1 to V6, and they have anatomical regions on the chest where they are being placed. Now, the V1 or C1, some electrode it is written as C1, on some is written as V1, whichever way. But the first one is V1, is placed at the 4th intercostal space. Now, and how do you get the 1st intercostal space? There's an anatomical landmark we use in ECG placement. We call this the sternal angle of Lewis. The sternal angle of Lewis, just at the edge of the mandible here. That's your 1st intercostal space. You count from there to your 4th intercostal space. Now, this is your sternum, breastbone or sternum. Now, at the edge of the sternum, so 4th intercostal space, right sternal border, we call it right sternal border. That's where you put your V1. Then to V2, the same 4th intercostal space, left sternal border, that's where you place V2. And before you place V3, you are going to place V4. V4 is along the mid clavicular line at the 5th intercostal space. Usually, the landmark is at just directly under the nipple. You see the 5th intercostal space there at the mid clavicular line. So after placing V4, then you now place V3, which is usually the halfway between V2 and V4. You place V3. Then V5 is the anterior axillary line. This is axillary, and the anterior axillary line at the 5th intercostal space. That's where you place V5. Now V6 is the mid axillary line. This is axillary too, then the middle of the armpit. This is the last edge of the body here, middle of the armpit, 5th intercostal space. That's where you place V6. Now for placing the limb leads, which are the color leads, we use color for them. This is how they look like. The limb leads. This is what we use for them. They are like clamps. So they have color. Some clamps will have their color on the rubber like this. Some will just have it here. There are 4 colors. We have yellow, red, and then green, black. Now between these four colors, the brightest colors goes to the upper limb, and the brightest colors are yellow and red. And between yellow and red, yellow is still the most bright. The brightest color is usually associated to the left side of the body. So the yellow color is attached to the left arm. Place the metal part facing the wrist. Now, I mentioned something about the ECG conductive gel. Before placing electrode on any of this region, you need to first swab the region to disinfect. Then after that, you add the ECG electrode for conductivity, for better conductivity. ECG electrode, you apply to the region. Then after that, you can attach either the chest lead or the limb leads. Now, then the red color goes to the right arm. The wrist part, the metal part goes inward to the wrist. Then we have green and black left. Between green and black, green is still brighter than black. So green goes to the left leg at the wrist part, at the ankle part, sorry, and then the black goes to the right leg at the ankle part. Now the ECG, after doing that, you can now connect the ECG electrode to the ECG machine. That's its own connector where you can connect to the machine. You start the machine, you take the recording, and then you print out your results. So that is that about ECG. Now let's talk about the procedure you need to take while taking this test, because I already described to you how you are going to do it whatsoever. Let's talk about the procedure, the things you need to do even before placing the electrodes. Firstly, you make sure that for males generally, most males have chest air. So what you do is you scrape the air, and this is to allow better conductivity so that the ears will not serve as insulator and causing artifacts. Then also make sure you properly untangle the wire. The ECG electrode wire is known to tangle easily. It tangles. After studying it and you bring it out, you see that the wires are tangled. You have to untangle it before placing the electrode, or else you will have artifacts. The wires will not be properly arranged and they will be causing artifacts in the results. Also make sure that you apply gel, the ECG gel. You swab the area and apply ECG gel before placing the electrode. Always make sure you apply ECG gel before placing the electrode. And once you are done with the procedure, and before that, before even placing the electrode, always tell your subjects to remove heavy metal in their body. Any form of metal, you talk about belt, bangles, wristwatch, make sure that they are all removed from the subject's body before they undergo the procedure. Then for patients that have artificial pacemaker, always inquire, ask them if they have artificial pacemaker, and then report to the physicians on ground in clinical settings. So they'll tell you, they can advise you further what to do for patients like that. And then for patients with amputation, maybe the hands, their limbs are amputated and you want to place electrode, there's a way they are also done. You use chest straps, form of disposable lips, electrodes that you can place along the shoulder here, which can represent the upper limbs, and for the lower limbs you can place them depending on the area of the amputation. You can place them at the upper part of the lower limbs. So that is that about ECG. Now let's talk about peak expiratory flow meter. Peak expiratory flow meter, I like that it is called in full, especially for the steeplechase, for the MBBS student. Always call the name in full: peak expiratory flow meter. The expiratory is important, the peak is important. So you get peak expiratory flow meter. And what is it used for? It is used for measuring the peak expiratory flow rate. Now, determining the peak expiratory flow rate can enable a physician to know if or suggest if an individual has an underlying lung disorder, whether obstruction or restriction, because in a normal individual there's a peak amount of force that can be exhaled from the lung. That is the peak expiration. Now with this apparatus we can determine the peak expiratory flow rate, and it is usually between 400 to 500 liter per minute for male and 350 to 450 for female. So this is the apparatus being used, and it is quite simple. Once you see it in your steeple shape, this is peak expiratory flow meter. Peak expiratory flow meter for determining the peak expiratory flow rate. Now we also have the under the respiratory physiology, we also have the spirometer. Spirometer. It's also an equipment used for the respiratory, for checking the respiratory parameters. So in the spirometric procedure, you can determine a lot of lung volumes and capacities. I think we've discussed that in the class. Yes, lung volumes and capacities. We discussed them in the class, and I have shown you the spirometer before. This is a spirometer kit. Here is the monitor. We have the respiratory pod. This is the respiratory pod. This is the disposable mouthpiece. And these are the power source. Because based on what we taught in class, I'm sure you guys will still remember how to operate the spirometer. So this is
Continue with urinalysis.
okay.
All those ones for 300.
This is the
So
nose piece. This is the nose piece. We just use it to hold the nose to avoid escape of gases, air, that we need in the spirometry test. So that is that for the 200 level revision. Let's talk about urinalysis. This is urinalysis. I like to call it urine analysis. That is urinalysis. Urine analysis to check the substance that can be seen in the urine, substance found in urine. Urine is a fluid and a waste product, but from this waste product, different tests can be carried out which can indicate the state of health of the individual. It can indicate whether some things are happening in the kidney, some things are happening in other parts of the body, because from that waste product, some of the substance of whatever metabolic activities that has occurred in the body can be found in that urine. The byproduct of those processes can be found in the urine. So from urine analysis or urinalysis, we can predict or detect some of these abnormalities. Now, in practical session, we use this urinalysis cup and the test strip to determine these abnormalities. So we have the urinalysis test strip inside the cup. This is a urinalysis test strip. It's a urinalysis test strip, and it is usually deep into urine sample. Once it's deep into urine sample, we wipe the excess urine up with cotton wool and place it on cotton wool for a minute. Then you can now check the color changes and compare it to the standards that is already made on this cup. Now, talking about the standards, we have different substance that can be present in urine, and the color changes that will occur on the strip that will detect the amount of this substance that is present in the urine. So depending on the color in which the strip matches with, you can tell us, okay, this is the percent of this particular substance in the urine. For example, blood can tell you, okay, this is the color change. With this color change, you know this is the amount of blood that is present in the urine. With this color change, you know this is the amount of bilirubin present in the urine. You have uroglobulin, you have ketone, you have glucose, protein, nitrite, leukocytes, that's white blood cell. You have the pH and the specific gravity of the urine sample. All you will need to do is just to compare the color changes to what we have in the urinalysis form. This procedure is very simple. It can be done by anybody. It's very simple. Even pregnant women or diabetes patients, they can have this, and just to check for the percent of protein and the glucose present in their urine, just an indicator of the state of health of that individual. So that is that about urinalysis. Now, aside that, there are other procedures we do in physiology practical that has to do with renal and renal physiology as a whole. We also have the urinometer. The urinalysis test strip can also determine the specific gravity of urine, but conventionally we use the urinometer to measure the specific gravity of urine. This is the urinometer. It's like in physics there's something we call Archimedes principle that has to do with buoyancy. So the way from the buoyancy of this tube in a urine sample, we can calculate the specific gravity of that urine sample. It has a gradation at the top here. So with the buoyancy, you can determine the specific gravity of the urine sample. So we also have the pH meter. With the pH meter, we have a pH meter in the lab. We have the pH meter to determine the pH and the temperature of the urine sample. Now, when we get a urine sample, aside from testing with the urinalysis test strip, from testing with the urinometer or with the pH meter, there are certain things you check for, when you see a urine sample, fresh out urine sample. What are the features you need to know? You need to check for the color, which can indicate the hydration or hydration level of the individual. You need to check for the smell. Normally, in an healthy individual, fresh urine is not supposed to smell. Just having your urine out, it's not supposed to smell until after it's left for some time and has turned to urea. It's now start to smell. And then you check for how clear the urine sample is. Is it clear or cloudy? The urine sample is supposed to be clear, translucent, where you can see through it. It's not supposed to be cloudy and muddy. And then the color, discuss about the color, whether it's clear, and the odor. Those are the things you look, the physical properties you have to look at when you get a urine sample before moving to testing it. So I think that is that for now. So let's discuss about welcome back. Let's discuss about GIT, the gastrointestinal system, the physiology of the gastrointestinal tract. There are some practical procedure we do is related to the physiology of the gastrointestinal tract. So here we are going to introduce you to some substance which you take, you ingest, individual ingest it, and you check for the salivary distribution and the rate of digestion of those substance. We have carbohydrates, which is usually represented with bread or yam. Then we have protein, which is we represent with meat in the lab. You have starch, you have sugar, you have We also use chewing gum to check for mechanical stimulus of saliva. So we have majorly what we do is the stimulants of saliva. So the first one is it can be given in the practical exam. We can give you chewing gum, which is a mechanical stimulant, to tell us and discuss about the salivary secretive phase while you eat chewing gum. The salivary secretive phase where you ingest bread, which that is salivary secretion that occurs where you take in carbohydrates. The salivary secretion that occurs with meat, which is a protein. And then we also did practicals on the pregnancy test, which is why we use the pregnancy test strip. And what we test for is the presence of ACG, human chorionic gonadotropin, which has its indicator in the pregnancy test strip. So if the ACG is present, the result will come out as positive, and if it's not present, it will be negative. That is, the individual is not pregnant. So we also did a practical on glucose tolerance test using the glucometer. So the glucose tolerance test is usually done with the fasting glucose tolerance test with time intervals. So those are the things you should be expecting. And then the glucometer, this is a glucometer. If you've seen it, it's a glucometer. Show you the glucometer and the glucometer test strip. This is a glucometer. This is a glucometer. And the test strip is present here. This is a glucometer. I could check glucometer. Now it is used to check for the glucose level in the blood. So things that is from here we come to the summary of the revision. See you next time.
So, guys, sorry that I stepped away. I went to do some official administrative work, and so my second had to take over. So, if you have any question and anything you want me to add to this video, please feel free to drop them in the comment section, and I will gladly do that. And if you haven't subscribed at this point, please make sure to subscribe, and I will see you guys